What do mitochondrial genetics tell us about human history? I'm here with Dr. Bryan Gitschlag, who is an evolutionary biologist specializing in mitochondrial genetics. So, welcome, Dr. Gitschlag. Thanks
Yeah,
for being here.
yeah. Thanks for inviting me. I would say mitochondrial genetics and theoretical population genetics. So, like the work I do isn't exclusive to mitochondrial genetics, but that's been the majority of my focus so far.
Awesome. Well, looking forward to what you're going to share today.
Sure. So, what I'd like to start by saying to kind of contextualize this is that there's some, I would say there's some confusion, uh, to put it one way, over like how quickly or at what rate does mitochondrial genetic diversity accumulate in the human population. And there are some studies that have looked at this or that have tried to estimate this that have been, in my experience, been passed around quite a bit without a lot of deep understanding of what the data are. And so I'd like to start by maybe providing just some basic principles of population genetics and evolutionary genetics that can help at least set the stage for understanding what it is we're looking at when we get down to the data.
Sounds
So
good.
it should be relatively straightforward, but again, please feel free to stop me if you have any questions. So the first thing that I'd like to start with is just covering a very rudimentary basic of how molecular sequences evolve. And the basic idea is that as the title, sorry, if you want to bring up the slides, that'd be great.
Mhm.
Okay, so I'd like to start with just some basic principles of how molecular sequences evolve. And so as the title of the slide here suggests, the evolution of molecular sequences is a process that combines the introduction of new variation and then the differential propagation through time of that variation. So if we start with a population where at a particular spot in their DNA, we have this sequence that we're looking at, GIG, GTG, excuse me. And then at some point along the way, there's an origin event of a new genetic variant, a mutation that flips one of those T's to a C. And then over time, it's possible, although it's not certain, that that C could spread through the population and become basically the new default variant or the new default nucleotide at that particular site, right? So this is basically the evolutionary process at the sequence level broken up into the stages of the origin of a new mutation and then the possible fixation of that mutation due to things like genetic drift or natural selection. You with me so far?
Yep.
Cool. So then what happens if, let's say, hypothetically speaking, in a different population, there's two different mutations that happen where that T gets flipped to a C, but then in someone else in that population, that T gets mutated to a G. And then over time, due to natural selection or genetic drift or some combination of the two, it's the G that actually becomes established as the new default. What the term that we would use is that the G goes to fixation, meaning that it's basically like an irreversible change at this point where you can't, because of natural selection and genetic drift, you can't flip back to everyone in the population having T again at that site like we did at the beginning of this timeline.
Mhm.
So fixation means like 100% of the members of the population now carry the G at that site. And then finally, we could conceptualize a third scenario where in one member, again, the T is mutated to a C, and in another member of the population, the T is mutated to an A. And then in this case, it's the C that goes to fixation at the expense of all the T's and the A's at that site in the DNA.
Mhm.
Okay, so if we're looking at these three scenarios as if they were three real populations, and we're drawing some conclusions on this basis, based on the information that's in front of us, one conclusion that we could draw is that the mutation rate from T to C is higher than the mutation rate from T to A or from T to G, because T to C happened 3 times, once in each population, and T to A and T to G each happened only once. However, we could conclude, at least again, based on the information in front of us, we might conclude that G has the greatest benefit on fitness because it was able to outcompete the C in population 2. However, C has a greater benefit on fitness than A or T because it was able to outcompete T or A when C was present with either of those two alleles. You with me so far?
Okay. Yeah.
Okay. So the basic idea that I want to sort of start off with is the idea that mutation rates can be broken down into a spectrum of mutation rates, where different kinds of mutations can have their own mutation rates. And then, of course, different mutations can have different effects on fitness. And so the probability of any particular type of mutation that could conceivably happen becoming an actual permanent substitution in that population is proportional to the probability that that mutation arises in the first place. So that's the probability of origin component of that of this formula you see in front of you, multiplied by the probability of fixation. So the idea is just that the probability of any substitution happening, it's just the probability that the mutation shows up, and then once it shows up, it's the probability that it also goes to fixation. That's a shorter way of viewing it down in the lower right. It's not important. Another way to think of this on a more conceptual, less mathematical level is that natural selection, when we're talking about different DNA molecules, like different copies of a DNA molecule floating around in a gene pool, one way to think of it is that natural selection decides what the winners are, but the mutation process decides who the players are. And there's these complicated probabilistic models that take into account these different forces of variation in mutation rates and variation in fitness effects, and then tries to predict the direction that a molecular sequence will evolve. And so that's kind of some of the theoretical work that I'm currently working on. Having said that, though, I'd like to shift gears to talk a little bit about some of the principles of mitochondrial genetics in particular, and some of the things that separate mitochondrial genetics from other forms of genetics. And so there's a few differences between mitochondrial DNA and the DNA of the nucleus. I presume that you and people listening may have heard of Mendelian genetics, or Gregor Mendel, sometimes called the father of genetics.
Yes.
And so Mendelian genetics refers to the genetics of the nucleus, right? So for example, if an individual with green eyes reproduces with an individual with brown eyes, and 100% of the offspring have brown eyes, then you can conclude on that basis that green is probably a recessive trait, right? And if you look at individual mutations in the genome, and you know that a particular mutation is recessive, and that and it's present along with like a dominant copy of the same gene, and that individual reproduces with someone else who carries both a recessive and a dominant copy of the same gene, then there's a 1 in 4 chance that any individual of the offspring generation will express the recessive trait, right? That's basically a kind of a quick and dirty overview of Mendelian genetics, at least the oversimplified version of it. Okay, Mendelian genetics doesn't really work when it comes to the mitochondrial genome. And one of the first reasons why is because mitochondrial DNA is what we call a high copy number genome. And so here we're showing a simplified visual of a cell where the large purple circle is the nucleus, and those little blue circles are copies of mitochondrial DNA that are dotted around the cell. And different types of cells have different copy numbers. So for example, some types of animal cell may have hundreds or even thousands of individual copies of the mitochondrial DNA molecule. And in fact, a mature ovum, right, like a mature egg, can carry over 100,000. It's been estimated over 100,000 individual copies of mitochondrial DNA. And here we're showing a simplified visual where there's 5 mitochondrial DNA copies. And so because of this high copy number because of this presence of high copy number in the cell, when a mutation happens, this mutation process gives rise to what we would call as a state of heteroplasmy. Hetero meaning different, plasmy referring to the cytoplasm. So it just means that there's different copies of mitochondrial DNA, like different non-identical copies of mitochondrial DNA that inhabit the same individual organism. Okay, so another difference between mitochondrial DNA and the DNA of the nucleus, other than the fact that it's present at high copy number, is that the replication of new copies of mitochondrial DNA is not tightly coupled to the cell cycle, right? So every time a cell divides, it makes a duplicate copy of the DNA in the nucleus so that each of the two daughter cells inherit one copy of the original DNA that was in the nucleus of the parent cell, right? But that basically means that the replication of new copies of DNA is coupled to the cell cycle so that the replication of new DNA happens once per cell cycle. The replication of new copies of mitochondrial DNA, however, happens throughout the cell cycle. And so over time within a cell, you can get variation in the heteroplasmy frequency. In other words, the percentage of copies of the mutated MtDNA within a cell, right? So the frequency at which a mutation is present, it can vary over time within a cell, and it can vary between different cells. And so this would be a heteroplasmic state. However, due to factors like natural selection, and in this case, it would be natural selection within a cell or within an organism rather than between organisms. So we're talking about natural selection at the level of some copies of mitochondrial DNA being able to replicate more successfully than other copies, perhaps depending on a mutation, for example. Or you can have genetic drift where just the frequency or the percentage at which a mutation is present within a cell may fluctuate randomly over time. And due to either selection or drift or a combination of the two, a mutation can go to fixation, not across an entire population, of course, but within an individual, right? And so now the cell that we're looking at over here on the right, that mutation has become a substitution, meaning that original mutated copy of mitochondrial DNA has gone to fixation, at least within this particular cell lineage. And so this is also something we would call a state of homoplasmy or a homoplasmic genotype, where there's only one variant or only one mitochondrial DNA variant within the cell. And so we might want to know, or we might want to try to account for, what are the sort of factors, what are the sort of mechanisms that can cause a mutated copy of mitochondrial DNA to either rise or fall in frequency, or even potentially go to fixation. And just as a quick aside, this was basically the focus of my doctoral dissertation work, is basically trying to fill in the gap. What is it that goes where that question mark is? What are these mechanisms that can take a mitochondrial DNA mutation and push it up to higher frequency within an organism?
Wow, perfect.
Cool. So I think that basically takes care of the gist of the overview that I wanted to share about mitochondrial genetics and how it's different from nuclear genetics. So if we're trying to apply this to human history, for example, if we want to estimate the time to the most recent common ancestor between two mitochondrial lineages, that's going to be a mathematical function of the number of substitution events that I was talking about on the previous slide, and specifically the number of substitution events that we would need to account for, which has to do with the number of differences in the sequences that we're comparing, and the substitution rate, right? The rate at which those genetic differences accumulate. And basically, if we take the substitutions and divide by the substitution rate, and the number of substitutions that we need to account for is basically half of the number of differences. And the reason is because if we're comparing two copies of mitochondrial DNA that descended from a common ancestor, they've both been accumulating differences on their own separate branches, right? And so the total number of differences between them is roughly going to be twice the number of differences that happened on either one of those two branches. Does that make sense?
No, I need more help on this. Okay. So What can you like? So what are we trying to do with the substitutions over substitution rate? Like how do you get the
So
like?
let's say you have two copies of mitochondrial DNA,
Okay.
and they differ at 10 sites, right?
Mhm.
So they're overall identical except for 10 spots in the DNA where they're different from each other.
Okay.
If you make the simplifying assumption that the substitution rate has been similar in the two branches, since they split from a common ancestor,
Okay.
then out of those 10 differences, 5 of them are due to substitutions that happened on branch A, and 5 of them are due to substitutions that happened on branch B.
I
So
see.
if you have
Okay.
a single ancestral copy of mitochondrial DNA and it becomes inherited by two different siblings, let's say, and then they go on to be inherited by the next generation and so on and so on. If this copy in one lineage racks up 5 substitutions and this copy in the other lineage racks up 5 substitutions, then that can result in a total of 10 differences between the two, even though there were only 5 actual changes that occurred in any one lineage.
Right. Okay. I got it. Mhm.
Cool. Okay. So the number of substitutions that we would need to account for is generally going to be half of the number of differences when we're comparing two sequences or when we're comparing any number of sequences.
Okay.
Okay, so, and then we would divide that by the substitution rate. So in this simplified example, let's say that there are 10 differences or 10 substitutions that we need to account for. And there's, on average, 0.1 substitutions per generation, or one substitution every 10 generations. And that comes out to 100 generations is the time to the most recent common ancestor for those two copies of mitochondrial DNA that we're comparing. Does that make sense?
Yes. Mm-hmm.
Okay, cool. Okay, so that seems simple enough. Excuse me. So now it's going to start to get a little bit more complicated, but
Okay.
please stop me if you have questions. So one factor that we need to account for, and as the title of the slide suggests, this is going to be a confounding factor if we're not careful in how we analyze the data, is something that we could call site heterogeneity in mutation rates. And this basically means a bias in the mutation rate among different sites in the DNA. And so I'll show you three examples. That's why the image that you're looking at is only confined to the upper left corner of the slide. So in the simplest case, there's no site heterogeneity in mutation rates, meaning there's no bias in the mutation rates. Every site in the DNA mutates with equal probability or with an equal rate,
Mm-hmm.
right? So you have one ancestral copy of DNA, and we're looking at a DNA sequence that's just represents 5 nucleotides. And on one branch, there's one mutation. On a second branch, there's a different mutation. And that adds up to 2 mutational events. And there's 2 variant sites, right?
Right.
So there's 2 sites in the DNA where they're different. And so the variation when we compare the 2 sequences is highlighted in yellow there. And the mutations are shown in red. The site where the mutation happened is shown in red.
Mm-hmm.
So this is a relatively simple, straightforward case because the number of mutational events corresponds to the number of places in the DNA where the 2 sequences are not identical. But then, because I'm wanting to kind of bring up the concept of bias in mutation rates between sites, another way to think of this is mutation hotspots. So let's consider what happens if you have a mutation hotspot. So first, in one generation, maybe you have a mutation that happens in one spot in the DNA and another mutation that happens elsewhere, just like we started with.
Mm-hmm.
But then very soon in a subsequent generation, you have another mutation that happens at the exact same site. And that can happen if that first spot in the DNA is a mutation hotspot, meaning it's more prone to mutating than other spots in the DNA.
Mm-hmm.
In this scenario, we have 3 mutational events because the same site mutated twice, and then there was a third mutation at a different site. But there's only one variant site, right? Because the first A in the beginning of that sequence mutated to a G, and then it mutated back to an A.
Yep.
And so the total number of differences between those two sequences is only one, even though there were 3 mutation events. You with me so far?
Yes, and
Okay.
this is great. You're a very good communicator. I'm happy for anybody who gets taught by you. So I hope people will really enjoy this.
I really appreciate that. Yeah. And again, please feel free to stop me if you have questions or if I start moving too fast. And I don't want to insult anybody's intelligence. I'm sure many people will know this, or maybe will be, maybe I'm going too slow because I'm trying to hammer the same point home. But I'm trying to speak to make sure that like everyone can understand and follow at the level of detail that I'm going to be talking about. Okay, so let's consider a third scenario where you still have a mutation hotspot, just like in this second scenario, but now it's a mutation hotspot that mutated in the same way on 2 independent branches. So in one lineage over here, you had a T to a C mutation, and then you also had a T to a C mutation in this other lineage over here. So we have a scenario where there were 2 mutation events, but there's no variant site. The genetic sequences are still identical when we, at least when we compare these 5 nucleotides.
Right.
So the main point that I want to emphasize here is that, just as the title of the slide suggests, this is a confounder, right? If you're measuring, if you're trying to measure mutation rates from human pedigrees, and some of the sites in the DNA are mutation hotspots, then you can actually observe a larger number of mutations than will actually be reflected when you compare the amount of genetic diversity among the individuals of the most recent generation. Does that make sense?
Wait, say it again. Well, let me tell you what's in my mind when I'm hearing this, and then maybe you can correct it if I'm thinking something wrong. Because like these 2 examples that you showed, the first example, which is straightforward, like 2 different mutations, so it's going to show up as, 2 variants. But then with these other ones, it's only going to show up as, 1 or none, even though there were more mutational events. But I don't like really understand how that would cause a problem for observing pedigrees. Like, yes, we wouldn't know accurately, but I don't know how that would mess up the mutation rate over time. I mean, like, of course, we would never see those, but I'm not sure how that would influence our mutation rate.
Sure. Okay. So I think one way of putting it is that the mutation rate, right, the rate at which a DNA molecule acquires a new mutation every time a copy of it is made, during reproduction, that mutation rate is not necessarily the same thing as the rate at which genetic diversity builds up. Because in these 2 examples on the right, the genetic diversity between these 2 sequences is either 1 variation or none, even though there were 3 mutations or 2, right? So in that first example, there were 2 mutations, there's 2 variant sites, right? So that everything's really simple. But then if you start to account for, or if you start to consider the idea that some spots in the DNA might be mutation hotspots, or they might have a higher average mutation rate,
Right.
then those mutations at those mutation hotspots, they might still be observed, right? Like you can compare the DNA of one individual to the DNA of their parents and say, okay, this site mutated. We observed a mutational event, but if that same site is mutating again, in a subsequent generation, or if a mutation at that same site is happening in a different lineage, then if you take the DNA of those descendants that have inherited the full number of mutations, you're going to see fewer differences between their DNA than is reflected in, you're going to see fewer differences in their DNA than the number of mutations that have happened.
Right. And I guess I can see for the middle one why that would mess up the pedigree if it was only 1 generation. right? Like, so if there's only one generation, then you're not going to basically, it'll be lost in the next generation. So you won't, I mean, that that could affect it. But what about the other one? Like, how would that affect? I mean, obviously it won't pick up that there was a separate event.
Mm-hmm.
But do you see what I'm getting at? Like, how would that like change the rate that we? I mean, it's not. We're not going to have the exact rate because we won't ever see it. But how would that mess up like the overall thing? Do you know what I mean? I'm not communicating as good as you are. That's all. You have to try to interpret what I'm saying.
Yeah. Okay. So let me see if I can. Okay. Let's put it this way. If you take the if you take the assumption that each of these arrows represents 2 generations, right? So maybe there's a hidden generation in between the top row of sequences and the second row of sequences. Then you can maybe infer that it takes 2 generations to build up 2 variant sites, right? Focusing on this first example.
Okay.
Okay. So let's say here's the data that we're looking at. The top, that pedigree, let's say is a mitochondrial DNA mutation rate pedigree. And down below is some measurement of the amount of mitochondrial diversity that exists when we compare 2 individuals. And so we can say, okay, there's 2 variant sites. It's been 2 generations within the pedigree. So it takes on average, 2 generations to build up 2 variant sites.
Okay.
Here, in this case, now there's one variant site, right? And there's actually been 4 generations. You with me? Because like there's the first row to the second row, that's the first 2 generations, just like in that first example.
Yeah.
And then from the second row to the third row, that last sequence, it's been another 2 generations. So now there's 1 variant site over 4 generations.
I see. Yeah. Okay.
So in that second example, you've had a longer amount of time, a larger number of generations to reach a point where there's actually only 1 variant site.
Right.
Compared to that first example where after 2 generations, there's 2 variant sites. So I guess the point I'm trying to make is that if you have a mutation hotspot, you can get a mutation rate that gives you a misleading estimate of how much variation to expect after a certain number of generations.
Okay. Yep.
In this third example, just like the first example, there's 2 mutational events after 2 generations, but there's no variation in the DNA when we compare the sequences. So depending on which of these 3 examples we're looking at, it could be 2 genetic differences after 2 generations. It could be no genetic differences after 2 generations, even though those 2 examples have the same mutation rate.
Right. Okay. Yep. I got it. Thank you.
Cool. Okay. Okay. So I guess I'll go ahead and close out this slide by saying that a big picture takeaway is that if you have different sites in the DNA that mutate at different rates, right, where the one that mutates at a higher rate we might call a mutation hotspot. It's often referred to as a mutation hotspot. That can drive a difference. Between the mutation rate and the actual per generation rate at which genetic differences build up.
Right.
So
Okay.
we're accustomed to thinking of, oh, well, the mutation rate, that's how quickly genetic differences build up. But that might not be the case. The mutation rate might be higher than the rate at which genetic differences build up, because
I
maybe those
see.
mutations
Okay.
are happening at the same site, or they're happening over and over again at the same site.
Got
Sorry,
it.
that is to say they might be happening at the same site in two lineages, or over and over again at the same site in one lineage, right? So the point being that the overall takeaway of this slide is that the mutation rate is not necessarily the rate at which genetic diversity builds up, right? Those can be different
Okay.
things because of hotspots.
Got it.
Okay, so turning to the real data then, this is all hypothetical. I haven't shown you any real data, so the question you might want to ask is, does this actually happen? And so I'd like to show some data from two studies that have been sort of shared around in creationism circles. One is this study by Howell et al. In the American Journal of Human Genetics, 1996. In page 504, they mention that they detected a T to C mutation at position 152. In the paper by Christina Santos et al. In the journal MBE, Molecular Biology and Evolution, 2005, they also reported what appears to be a T to C mutation at position 52. There's a little, you can see there's a little question mark because it's not 100% clear which direction that mutation happened, whether it was T to C or whether it was C to T. However, the state in homoplasmic individuals is T. The consensus sequence outside of Africa is T. And so it's looking like it was a T to C mutation rather than the other way around. If it was the other way around, though, that would just mean that it's a reverse of an earlier mutation that was T to C. So
Right.
in any case, there appears to be a T to C mutation that happened at the same position, at position 152, in these two different families. And so, granted, there's other mutations that are happening, but if we ignore those other mutations for the sake of argument, and we're just comparing the site here, there's two whole mutations that have happened without contributing to genetic differences between these two families. Does that make sense?
What do you mean not causing differences between the? Okay, because you're saying, oh, it's two different events,
it's that third
Okay.
scenario over here to the right.
Yes. Okay.
Okay. Moving on, again, the Howell paper, they also report a T to C mutation at position 195, and the Heyer et al. American Journal of Human Genetics 2001 paper also reported a T to C mutation at position 195. And again, it's just like this, it's just like this third scenario. I'm going to show you a few examples from the literature of this third scenario where you had the same mutation happen in two different lineages independently without actually changing the number of differences between those two lineages.
And were these synonymous mutations or non-synonymous?
These are neither, because this is a non-coding region, and synonymous and non-synonymous refers to mutations that happen in a region that codes for DNA, or sorry, codes for amino acids.
Oh, got it. Okay, yeah, cool.
Yeah. So there's the T to C that was reported as a mutational event in Howell et al., and another one at the same site in Heyer et al. In the paper by Parsons, there was actually a G to A and a to G mutation at the same site in two different individuals in I believe that's the Armed Forces DNA Identification Laboratory. I think that's what AFDIL stands for. In any case, of the DNA substitutions that were reported in this Parsons paper in Nature Genetics 1997, two mutations happened at the exact same site. And now those are mutations in different directions, right? One goes from a G to A and the other one goes from an A to G. But the point is that those are not actually adding to the number of differences between two individuals. Does that make sense?
Yes.
Okay. Also sticking with Parsons, there's a mutation at position 16093 that goes from T to C. And in this other paper by, I believe it's pronounced Sigurd Ardóttir, in the American Journal of Human Genetics 2000, there's another substitution from T to C at position 16093. So again, it's just like that third scenario, that third hypothetical scenario I showed, where you have the same mutation at the same site in two different families. And so if we're just focusing in on those mutations in particular, there's these mutations are happening, but they are not actually contributing to the growing number of genetic differences between individuals.
Cool.
Okay. And then finally, there's a mutation at position 16182. This was recorded as either an A to C or a deletion of A. And over here to the right, sorry, this first table here was also in the paper by Cristina Santos et al. In MBE, Molecular Biology and Evolution, 2005. The second table, I'm showing you one column from table 1 of Madrigal et al., Madrigal and colleagues in the American Journal of Physical Anthropology, 2012. Now you see in that red box the number 182. There's a footnote down there that says nucleotide position minus 16,000. So it's actually 16,182. You subtract 16,000, that's where the 182 is coming from. So the point is that this is a mutation that happened at the same site in the mitochondrial DNA as this other red boxed mutation in this table by Santos et al. And also sticking with Madrigal et al., they actually report, citing earlier literature, that some of these other sites that are shown in red boxes here are mutation hotspots. They also mention that they suspect positions 182, 311, and 362, that top red box, they annotate that as a suspected adoption because it's an unusually large number of differences to have happened within one family. And so one way to explain that is an adoption. Another way to explain that is that this is a complete statistical anomaly. But in any case, this is not reflective of what is typically measured as the mutation rate. And then also they mentioned that there's a poly-C, meaning a stretch of the DNA nucleotide cytosine, CCCC. There's a poly-C repeat that can cause genotyping errors and sequencing errors. And so it's possible, we don't know for sure, but it is possible that that position 182 was misidentified as a mutation. And they take into account these uncertainties, because what they do in this paper by Madrigal et al. Is that they provide basically a range of different options for what the substitution rate might be, depending on whether they include these sites that might have been sequencing errors or not, that type of thing. Okay, so just going back to this slide, there's a few different examples. There's the T to C at position 152. There's a G to A and an A to G at position 207. There's a T to C at position 195 and a T to C at position 16093 that are reported by these different papers that are shown in the bottom right. And so again, the main take-home message that I want to emphasize is that if you have sites that are mutation hotspots, where the same mutation is happening multiple times in a lineage, or it's happening at the same site independently in two different lineages, then those mutations they're happening, like those are real mutations, but they're not actually contributing to driving up the amount of genetic diversity between those different lineages in which those mutations are happening.
Right.
Okay, so that's the first take home that I wanted to mention. So maybe I would say I would advise you to maybe file this in your short-term memory, because I'll be moving on to a different confounding factor. So the first confounding factor is site heterogeneity in mutation rates. In other words, mutation bias between sites, right? In other words, mutation hotspots. So that's a confounder. Another confounder is tissue type. And there's multiple different tissues, right? There's the tissues that are reproductively play a direct role in reproducing, right? The sperm and the egg, and those we call germline tissues, and also the cells that give rise to germline to the sperm and egg, right? So the cells that are dividing early in embryonic development that are destined to become that are destined to pass their DNA into what will later on be a mature sperm or egg, those are all part of the germline. And then all the other cell lineages that they don't pass their DNA onto the next generation, those genetic lines die when the body dies. Those are what we call somatic tissues, right? So muscle, blood cells, different tissues that make up different organs, neurons, everything that you see over to the right half of this image, basically. Okay, so why is this a confounding factor in mitochondrial genetics? So let's take the difference between these two types of tissues and simplify it down to a very simple hypothetical organism that has a soma, a body, right? The somatic tissue highlighted in yellow, and germline shown in kind of a rust color. There's a process of reproduction where this organism contributes one gamete from its germline, and over the course of development, that gamete, if it becomes fertilized in the case of sexual reproduction, will develop into the next generation of organism, right? And let's populate that organism with a simplified down number of mitochondrial DNA. So these little blue circles, can you see them okay?
Yeah.
Okay. And so we have a development process also where there's intermediate stages of development between a single-celled fertilized egg and an adult body. And so we could ask what happens if there's a mutation in the mitochondrial DNA in the somatic tissue, right? One of those yellow cells. And here I'm showing the reference or the unmutated copy of mitochondrial DNA in blue and the mutated copy of mitochondrial DNA in red. And so what's going to happen is that as those cells of the soma grow and divide, they can populate the adult body with mutated copies of mitochondrial DNA, but those are going to be confined to the somatic cells of that organism. And so in the next generation, assuming that organism is unaffected by this mutation and they're still able to reproduce, the next generation is going to inherit only the unmutated copies of mitochondrial DNA. You're with me so far?
Yes.
Cool.
So this is like granddaughter over here. It would be because of the mutation in the somatic cell of the mother, the daughter could have the mutation in the somatic cells, but then it wouldn't go further than that because it wasn't actually in the germline. So it would not be in the granddaughter.
That's right.
Okay.
Okay, but then we could ask the hypothetical question of what happens if there's a mutation to the mitochondrial DNA in the germline of the grandmother. Well, that mutation can then spread to the next generation and the next one and so on, because the mitochondrial DNA, what it needs is an unbroken lineage of cells to be able to pass from one generation to the next. And that unbroken lineage of cells is what we call the germline. So if that mutated copy of mitochondrial DNA happens in the germline, then that mutation can potentially, it's not certain, it can potentially get passed on to future generations. And you're with me so far?
But I'm wondering, why wouldn't it be certain? Is it because the grandmother is heteroplasmic, so it might not the one mutation might not pass over?
Well, it might get selected against, right? Like the natural selection at the micro scale. Micro, I realize is a term that is very easily confused, or very easily confusing in this context. What I mean is at the subcellular scale, right? At a very, at the scale of different copies of mitochondrial DNA within a cell, natural selection might disfavor the mutation, and it might favor the unmutated copies of mitochondrial DNA, so that maybe only the unmutated copies of mitochondrial DNA actually survive to the next generation, even though that mutation happened in the germline. And we do in fact know that there are mechanisms in the germline that can decrease the likelihood of a mutation getting passed on to the next generation. And that depends on the effect of the mutation. It depends on where in the mitochondrial genome that mutation happened and what its effects are. But that is something that can happen.
Now, this brings me to I know this is totally interrupting your presentation here, but
You're fine.
I want to grab onto it because this is what I read in your paper that you did. And I think you were studying roundworms and the mitochondrial pass on. And like, I think you said like the mutant that like is maybe even like slightly deleterious could still get passed on and selected for.
Yep.
Is that, can you explain more about that? Like what you learned?
So I have to warn you, this
deep?
is going to be a
Too
huge
deep?
tangent. Excuse me. But yeah, absolutely.
Should we save it? Should we save it for another conversation then? Or is it relevant? Is it?
I can give you a short version.
Okay.
So so on one level, it depends. But one of the things we found, and one of the things that I found that's kind of like an accumulating, I don't want to say discovery because that makes it sound like an individual event, but it's like something that's over time becoming increasingly clear in the field as more research comes out, is that a mutated copy of mitochondrial DNA can proliferate, where its numbers increase over the course of development as the animal develops. And then as that animal goes to reproduce, the numbers or the allele frequency, right, the percentage of copies of mitochondrial DNA in that organism that carry the mutation, that frequency can actually diminish as the pool of mitochondrial DNA is being passed from the adult germline into like an individual oocyte or an individual ovum. And so what we found, like one thing that I found in my 2020 paper in the journal eLife, and it's also been replicated elsewhere by other independent studies, is that if you compare the allele frequency of a mitochondrial mutation between an adult parent and between the eggs, right, the ova that come from that adult parent, you can actually see the mutant frequency declines. And so it's a statistical trend. So it looks like it's not just randomly happening, but that there's something that is selecting for the wild type, the unmutated copy of mitochondrial DNA over or at the expense of the mutated copy. However, if that mutation survives, meaning as long as its frequency doesn't drop to zero, then as that ovum develops into the next generation of adult, it can rebound and the frequency of that mutation can rise again. And in fact, it can rise so much that the adult of the next generation carries that mutation at an even higher frequency than the original parent did. So that's the short version.
Wow. Okay, cool.
And again, it depends on the mutation. It depends on the effects of that mutation, where it happens in the germline, or sorry, not where it happens in the germline, where it happens in the genome. So like one of the big takeaways of my most recent mitochondrial study is that we see different evolutionary mechanisms, so different modes of selection and different effects of genetic drift affecting a mutation depending on the nature of that mutation, you know, which gene is affected and where that mutation happens in the mitochondrial genome.
Cool. Okay. I'm ready to keep going if you want to.
Sure, yeah. Okay, so the reason I wanted to share all of this is because I wanted to drive home the point that you need an immortal cell lineage, right, that can pass its DNA from one generation to the next and so on, in order for that mitochondrial mutation to continue to stick around in the population, right? If that mutation is confined to the somatic tissue, it'll fizzle out. It'll disappear from the population by the next generation. And so if we look at a human pedigree study of mitochondrial mutations, and we see, okay, in the parent generation, there's only the unmutated copy of mitochondrial DNA, but in the next generation, there's a mutation. Then we can look at that and say, okay, look, a mutation happened. Do we count that mutation or not if we're trying to do a study that estimates the mutation rate? Well, it depends, because if we're trying to do a study that estimates the long-term rate at which genetic diversity increases in the population, then we need to be very careful about identifying for each mutation, is that a somatic mutation that's confined to the somatic tissue and it's going to be gone by the next generation, or is it a germline mutation? And in some cases, in some studies, it's not actually clear, because a mutation might be reported because it's present and it wasn't present in the parents, but we don't know in which tissue did that mutation first arise. Does that make sense?
Yes.
Cool. And so we might want to know, okay, just like in the case of hotspots, this is nice and hypothetical, but are there cases where this actually happens? And it appears to be the case, or at least I should say that we can't confidently, in the case of some studies, we cannot confidently determine whether or not that mutation we can't pin down which tissue that mutation happened in, basically. And so here, what we're looking at is two pedigrees that are shown from Figure 1 in that study that I mentioned earlier by Christina Santos et al. In the journal Molecular Biology and Evolution 2005. The black arrows that you're looking at are individuals in which a mutation was identified. If you don't see an arrow, there was no mutation identified. And so what we're looking at here is cases like this, where there's no mutation in the parent, at least no mutation that was identified in the parent, but there was a mutation identified in the offspring. And so we're looking at cases like that, and that's basically what we're seeing here. So individuals that have a mutation that wasn't in the parent, but we can't tell if that mutation, we can't tell what tissue that mutation happened in. So we cannot be confident that that mutation is in the germline, and therefore we cannot be confident that these mutations that these arrows are pointing at are going to get passed on to future generations. Okay. And that's something that we need to be careful of in how we estimate mutation rates, right? Because if we're trying to estimate the long-term rate at which genetic diversity accumulates, and we can't, we can't be clear that we're dealing with germline mutations, then it's kind of an open question about the data, and we can't be certain whether or not these mutations should be counted in our estimate of that long-term substitution rate or not. And that uncertainty is actually something that was part of the purpose of this study by Christina Santos et al., right? They're trying to account for these types of things that can happen that can prevent a mutation from getting passed on, right? Whether it doesn't happen in a tissue that it doesn't happen in the germline, right? Or there's other factors that I'll get to in a moment. But that was part of the point of this study. You with me so far?
Yeah.
Cool. And then finally, we need to take into account, well, I say finally, there's a couple of other things I want to get to, but we need to take into account the mode of inheritance. So mitochondrial DNA is maternally inherited, right? And so what that means is that in order for this mutated copy of mitochondrial DNA to continue to get passed on to future generations indefinitely, you need an unbroken mother-daughter lineage, right? And so mitochondrial DNA, it's maternally inherited. It's basically male members of the population are not going to pass on their DNA, their mitochondrial DNA, to the next generation. It can happen on rare occasions, but again, it is very rare. And when it does happen, it's also usually something else that is that has either gone wrong or has perhaps been affected, right? So there are some studies that report what we call paternal inheritance, where an individual has inherited mitochondrial DNA from their father. But some of these studies that report this, the way they were able to identify it in the first place is because a patient is being seen by medical professionals for a mitochondrial disease, right? And so it's usually an indicator that something has gone wrong. The overwhelming majority of the time, mitochondrial DNA is only maternally inherited.
I see.
Cool. So then what happens if you have a mother who has a mitochondrial mutation, but she only has male children, that mutation is not going to get passed on. Again, except in very, very rare cases. Or what happens if a mother has a mitochondrial mutation, but she only passes that mutation on to her male children, not to her daughters. Again, that mutation is not going to get passed on. And so the overall takeaway, the third takeaway, the first one was hotspots. The second one was which tissue is the mutation happening in, germline or soma. And the third takeaway here that I want to emphasize is that it matters the mode of inheritance. It's something that we need to take account for. And so you need, again, you need an unbroken mother-daughter lineage in order for a mitochondrial mutation to get passed on indefinitely. And so we can, again, ask ourselves, if we look closely at the data, is this a confounding factor that it actually affects some of the estimates that people have concluded from these studies? And the short answer is yes. So the example over on the left, I'll go ahead and orient you to that if I could first. This is an Amish family represented in figure 2 from the Parsons et al. Nature Genetics paper from 1997. And basically what you're looking at is blue represents the unmutated copy of mitochondrial DNA, just like the color coding that I've been using in my own animations. Red represents the mutated copy of mitochondrial DNA. Purple represents an individual who is heteroplasmic, so they contain a mix of the mutated and unmutated copies of mitochondrial DNA. And the circles are female individuals and the squares are male individuals. And so even though this is one of the mutations that was counted in the Parsons et al. Study, it was only passed on to a male member of the family. The female siblings did not inherit that mutation. And so we're dealing with this example, like this example that I'm showing over here to the right, to the far right. So this this hypothetical example is something that we see in the data when we look at figure 2 of Parsons. So there's a mutation that really shouldn't have been included if they were trying to estimate a long-term buildup of genetic diversity, because this is only a male member of the family that inherited that mutation, and so it's not going to get passed on to the next generation. Okay, and then the other example was figure 1 from Sigurdardóttir et al. In the American Journal of Human Genetics, 2000. And so in figure 1, they show a bunch of pedigrees, and in this particular one that we're looking at, the individual that is shown in light blue, kind of a grayish blue, if you look at the figure legend of figure 1 in this paper, they mentioned that individuals highlighted in light blue are heteroplasmic for a mutation. And so in this case, we have a mutation that's only detected in a male member of the family.
Okay. So why do you think these guys are counting that if they're like trying to make a mutation rate that can be extrapolated back into the past?
Mm-hmm.
What is their thinking here?
That's a good question. It's possible that this took some time to figure out that mutations are oftentimes not being passed on to daughters, right? You need a mutation that's passed on to daughters in order for that mutation to get passed on to the next generation and the next one, right? And so you have a lot of heterogeneity in reproduction among different families where, if I go back to this slide, bear with me, sorry, while I walk you back through my animations again. But you need this scenario on the left for that mutation to be passed on indefinitely.
Yeah.
These two scenarios on the right are not going to cut it, right? And this is a conclusion that we can say now in 2025 with the benefit of hindsight and looking at decades worth of research. But it's possible that it's a consideration that wasn't thought through very rigorously at the very start of when they started doing mitochondrial genetics, right? The first study that I'm aware of, that I'm not saying the only study, but the first study I'm aware of where they really were careful and explicitly tried to account for this issue of maternal inheritance, and that you don't count mutations if they're only inherited by males. The first study to really explicitly account for that I'm aware of, at least the first one that's on my radar, is the Christina Santos et al. Study in 2005.
Okay.
And so that would be the one over here on the right.
Okay.
So yeah, so you're with me so far?
Yeah.
Cool. And then finally, there's one other confounder. I knew there was another one that I had forgot about, which is the presence of heteroplasmy, right? The fact that a single cell might contain a mix of the mutated and unmutated copies of mitochondrial DNA. And so what you're looking at is, sorry, I should have labeled the axes, but there is kind of a label up at the top there. But the vertical axis is the probability that a mutation goes to fixation, right? So it can be from 0 to 1. And the horizontal axis is the allele frequency, right? So that's the number of mutated copies of DNA divided by the total number of copies of DNA within whatever sample pool we're looking at. And so there's different colored curves that correspond to different modes of selection. And you keep seeing the symbol show up on this graph, NS. So S is the selection coefficient. So that's just the effect that a mutation has on fitness. And N is a measure of the effective population size. So N times S, if that number is if the fitness effect times the population size is greater than 0, then that's a beneficial mutation. And that's why you have these curves that start more steep and then they level off, because you don't have to, if it's a beneficial mutation, it doesn't have to rise very high in frequency before its probability of going to fixation really goes up. If it's a deleterious mutation, that are those lower curves, right? And so the population size times the selection coefficient is below 0. If it's a neutral mutation, if it's a perfectly neutral mutation, then you fall right on that red diagonal line. And so here, because all of the studies that I've talked about so far have focused on the non-coding region, and most of them have focused on what's called the hypervariable region, where there's the part of the mitochondrial genome that has the greatest amount of sequence variation in the population. And so those studies focus on that region, partially because it is thought that mutations there tend to not have much of an effect on fitness, and therefore it's thought that natural selection isn't really a factor. And so if we make the simplifying assumption that most of these studies make, that these mutations are generally neutral, then the probability when you're in a heteroplasmic state where an individual organism or an individual germline cell carries a mixed population of mutant and wild-type mitochondrial DNA, then the probability that that mutation rises to 100% allele frequency within that lineage is whatever the existing allele frequency, whatever the current allele frequency is, right? So if that mutation is present at an allele frequency of 0.5, 50%, meaning half the copies of mitochondrial DNA in that organism are mutant and the other half are wild type, then it's the probability that the mutation goes to fixation, 100% frequency, is basically 50-50, because it's equally probable that the wild type mitochondrial DNA could go to fixation, or the unmutated copy of mitochondrial DNA could go to fixation.
Cool.
Did that make sense?
Yes.
Cool. Okay, so one of the last things here that study that I mentioned, Christina Santos et al. Tried to account for is the probability of fixation, at which, just like I mentioned and I'm showing here, that's a function of whatever the frequency of that heteroplasmic mutation is. So if we take a closer look at the data from this study, here we're looking at table 3. There's a few deletions. There's a couple deletions, but if we're just concerning ourselves with the single nucleotide substitutions, where a T is mutated to a C, for example, or an A to a G, then there's 11, right? They report 11 single nucleotide replacement type mutations. And in table 2, I'm not showing it here, but I'll show the data in a second. In table 2, they report the heteroplasmic frequencies for at least some of these. So the raw data is, by the way, I should say they're looking at a sequence window of I believe it was 946 nucleotides. And so they count 11 substitutions. There's 946 sites. And the study looked at 321 generations worth of human pedigree data. And if you do the math, that comes out to roughly 3.68 times 10 to the minus 5 substitutions per site in the DNA per generation. However, these two mutations that I'm pointing at in red are not going to be transmitted to daughters. If you look closely at the pedigrees, those are carried by male individuals in the family. There's 4 individual types of mutation that some of them are carried by different members of the family at different heteroplasmic frequencies. So for example, the mutation at position 16150, the second row, that was detected at a frequency of 0.213. 21% of the mitochondrial DNA in that individual, roughly, was the mutated copy. And then,
so it's probably not going to stick around. More likely not to stick around.
It might, but there's multiple mutations here, right? So they add up to some non-zero amount that at least some of them are going to stick around.
Okay.
And so down below, for example, there's a C to T mutation at position 150. There's that mutation T to C, I believe, is most probable at position 152, and then finally that final mutation looks like a A to G, although I don't think it's completely certain, at position 215. But they report the heteroplasmic frequencies of what they score as the mutated allele with these numbers. So the mutation at position 150, for example, 2 individuals carry it, 1 person at 2% frequency, the other person at 4% frequency. And you can see the other heteroplasmic frequencies, position 152, there's a person that carries the mutated copy at 25% frequency and the other person at 31% frequency. And then that last mutation, someone carries it at 33% frequency and someone else carries it at 13.8% frequency. And if you add up all of those frequencies, using basically accounting for the probability that some of these, but not all, are going to go to fixation in a way that's proportional to the allele frequencies that they're present at, just like that graph I showed on the previous slide, then they estimate that 1.308 actual substitutions per 946 sites per 321 generations. And that comes out to 4.31 times 10 to the minus 6 substitutions per site per generation. And this is based on the idea that they're also trying to account for tissue type, right? Remember, I said earlier that if a mutation happens in the soma, it's not going to get passed on. A mutation has to be a germline mutation. So in this table, we're seeing a few mutations that are scored as not being germline mutations. And that's based on the idea that a mutation was only observed, for example, in a most recent member of the family, but it was not observed in their offspring, or that person didn't have offspring, and the mutation wasn't observed in the parents. And so we can't be sure that these mutations are germline mutations. But if we assume that they are, there's 2 additional mutations that we could include. There's the other ones we wouldn't include because they're either definitively not present in the next generation, or they are only present in male members of the family. But for the mutations that if they are germline mutations, that they have a shot at getting passed on to future generations, there's 2 other ones that we could include. One is at a frequency of 0.577, 57.7% mutant MtDNA in that individual. And then there's another mutation that's present at 25% frequency. And if you take these into account, then it bumps it up from 1.3 to 2.135 substitutions per the 946 sites in the DNA that they're looking at per 321 generations. And that comes out to about 7.03 times 10 to the minus 6 substitutions per site per generation.
And what does that in simple terms come out to? Do you have it available? Like how many per generation?
Yeah, so that is what these numbers are. So,
yeah, no, I mean like how many generations does it take to get one?
Yeah, I mean we could work that out. We'd have to take a side quest to work that out. It's basically just 1 divided by these numbers down below. But we can get to that in just a moment. But accounting for these various confounding factors, again, those confounding factors are that some mutations might not get passed on because they're present in the somatic tissue, or some mutations might be passed on because they're germline. Some might not be passed on because they're in the soma. Some mutations won't get passed on if they're not inherited by female offspring, right? So if a mother only has sons rather than daughters, or if she only passes the mutation on to her sons, then that mutation is not going to get passed on. And then finally, a mutation may or may not go to fixation. It may or may not become a permanent substitution within that genealogical mother-daughter lineage, depending on the heteroplasmic frequency, right? And so taking those factors into account, we can ask, how much do these short-term pedigree studies, if they were not to take into account these different factors, how much would they be overestimating the rate at which mitochondrial DNA diversity accumulates over time? And the simple, that question, the answer to that question, the simple overestimate would be the rate, the raw rate for those mutations divided by the rate accounting for sex and the probability of fixation. In other words, 3.68 times 10 to the minus 5 substitutions per site per generation over, again, that estimate, depending on whether or not those uncertain mutations, depending on whether or not we count them as somatic or germline. That gives us a range of estimates from 4.31 to 7.03 times 10 to the minus 6. And that comes out to a 5 to 8-fold. So in other words, to bring it all home and kind of wrap up this discussion of this study, they found 11 substitutions in 321 generations worth of data. But there's these confounding factors I mentioned, where a mutation might have been in the soma rather than the germline. It might not be inherited in a direct, unbroken mother-daughter lineage, and it might be present at a heteroplasmic frequency that gives it only a marginal probability of becoming a permanent substitution. And so when you account for these factors, the raw unaccounted for rate overestimates the substitution rate by about 5 to 8-fold.
Okay, but I need to get to the numbers on that are not, in scientific notation. Like I need to know what this actually comes out to.
Sorry, could you clarify maybe which number you're thinking of?
Okay, what my understanding is that under evolutionary expectations, before we started doing all these pedigree studies and finding out what the mutation rate is or substitution rate is, if if that's the term I should use, is that we would expect that it would be 1 mutation for every 600 generations. And so I'm wanting to know, and then, what, the papers back in the late 90s and stuff we're showing was something more like 1 in 30, 1 in 33 generations. So I'm trying to figure out what is the number that, Santos got accounting for the factors that you mentioned.
Okay, if we are ignoring hotspots, for example, and we just assume that the 946 DNA nucleotides that they looked at all have an equal probability of mutating, then somewhere within that entire sequence window of 946 nucleotides, you would expect a new mutation to happen on average roughly somewhere between 1 and 150 generations to 1 and 245 generations. Let's round it off for simplicity's sake, somewhere between 150 to 250 generations go by for each new mutation that happens. That range, again, is dependent on whether or not we count these uncertain mutations as being somatic or germline.
Okay. Okay, so would you say that resolves the issue of the difference between the pedigree and phylogenetic studies?
Not completely, because one thing that this study did not account for, or didn't like explicitly, let me back up. They refined their estimate of the substitution rate by explicitly accounting for those three factors that I mentioned of heteroplasmic frequency that gives you an uncertain but estimatable probability of fixation, the tissue that the mutation happens in, somatic or germline, and the sex of the lineages that those mutations are present in, specifically if a mutation is only inherited by males or if it's present in an unbroken mother-daughter lineage, right? Accounting for those explicit factors, they found that the raw mutation rate of just 11 per 321 generations overestimates the more rigorous, more refined rate by a factor of about 5 to 8-fold. But one thing that estimate does not explicitly account for is the confounding factor of hotspot mutations, which was that issue that I first started with. And so we would have to dig into the data a little bit more and try to continually refine these estimates. And one thing that more recent and ongoing research in mitochondrial genetics has tried to do is expand the scope of these studies by including not just the hypervariable regions or not just the non-coding region, but the entire mitochondrial genome. And it makes things a little bit more complicated, but because you have the issue of a big confounding issue that we now have to account for if we're going to look at the entire mitochondrial genome would be natural selection, because a mutation that happens in a protein coding gene is much more likely to have an effect on fitness than a mutation that happens in the hypervariable non-coding region. So there's a trade-off where it becomes a bit more complicated because you now have to account for selection. But the good news is we have data that tells us something about how selection works in the mitochondrial genome. And so the trade-off is that we have more data to work with, and we can start to refine these estimates by taking into account the entire mitochondrial genome and taking into account how selection works.
Okay, so do you believe overall that's the preferred method to take the entire? Mhm.
Absolutely. Yeah, I mean, you have more data to work with, and we like I said, there's data that tells us how natural selection works on the mitochondrial genome. So some studies, especially earlier mitochondrial genetic studies from 15, 20, 30 years ago, this study by Santos et al. Was from 20 years ago now. Actually, I can't believe I'm saying that. 2005 was 20 years ago. But some of these earlier studies from 20, 30 years ago, they were looking at mutations within that narrowly defined region. It's less than 10% of the total mitochondrial genome, right? That is non-coding. And part of the benefit of that, again, is that it was thought, at least, that region provides a much more regular molecular clock, because without having to account for natural selection, it was thought that the rate at which genetic diversity builds up is going to be directly given to us by the mutation rate. And that's not necessarily the case for all of these reasons that we've been talking about. However, we can take a look at the data. There's data available that we can take a look at that pertains to the long-term substitution rate across the entire mitochondrial genome. And of course, that the mutation rate is much higher in the hypervariable region than it is in the protein coding regions.
Right.
But that's okay. We have data that can tell us how how quickly genetic diversity builds up across the mitochondrial genome, even in the protein coding regions, especially when we account for selection.
Okay, so when they do the one for the whole mitochondrial DNA, are they taking into account all the differences in all the regions? Because from my understanding, there's like great differences in the way that, like you said, there's hotspots and things like that. So they're trying to take into account all of those factors.
Yeah, and I mean, that's a great question, and it's absolutely true that there is heterogeneity of mutation rates at different sites throughout the mitochondrial genome, and that it's something that is important to take into account. And that's something that studies do take into account, especially as the field of mitochondrial genetics has progressed and gotten more mature. The models that have been used to study the history of the human mitochondrial genome have become more sophisticated, because the models have like mathematical parameters that can take into account things like different types of mutations happen at different rates, different sites in the genome happen at different rates, and there's models that can account for that.